Hello, this is Dr. Rina Rani Ray. Welcome back to the second part of structure of bacteria. We know the outermost layer of the bacterial cell is a capsule or slime layer. It is made up of glycocalyx. Glyco means sugar, calyx means cup. So it is made up of polysaccharide. It helps the bacteria to adhere on the surface. As for example, S mutans adheres on the enamel of teeth with the help of this glycocalyx. It prevents phagocytosis because complements cannot penetrate the sugars. It prevents desiccation. It protects the bacterial cell from antibiotics and antimicrobials, and it helps during the starvation because it can be consumed as a sugar. Capsule is a condensed layer, closely surrounds the bacterium, and it cannot be washed very easily. Whereas slime layer is loosely adherent, non-uniform in density and thickness, and it can be removed quite easily. So both capsule and slime layer are called glycocalyx. Now regarding the cell wall, it's made up of peptidoglycan polymer, the amino acids and sugars. It is the unique feature of the bacteria. The sugars are N-acetyl glucosamine and N-acetyl muramic acid. They are called NAG and NAM respectively. The D form of amino acids are used, not the L form, because the D form is very hard to break. This amino acids cross-link NAG and NAM, and how they are cross-linking we are going to discuss later on. Now about the functions of cell wall. Cell wall determines the shape of the bacteria. The cell wall prevents osmotic shock and physical damage. It regulates the transport of material in and out of the bacteria. It helps in movement of the cells. It takes part in protein synthesis, and some antibiotics affects directly like penicillin, and it also helps in mineral storage. Now, depending on the amount of peptidoglycan present in the cell wall, we know the bacteria can be divided into gram-positive and gram-negative. Gram-positive bacteria have walls containing relatively large amounts of peptidoglycan, and so they can retain more amount of crystal violet. They are having teichoic acid also. This teichoic acid is a unique feature of gram-positive bacteria. As for example, Staphylococcus epidermidis, Streptococcus pyogenes, and Clostridium tetani, all these are the examples of common gram-positive bacteria. On the other hand, gram-negative species have walls containing small amounts of peptidoglycan and a lipopolysaccharide, that is a fat sugar combo. These are harder to control with antibiotics, and these lipopolysaccharides are a unique feature of gram-negative bacteria. The common examples are E. coli, Salmonella typhi, Vibrio cholerae, etc. Now we are going to concentrate on cell wall of gram-positive bacteria. Here we can see the schematic representation of the cell wall of a gram-positive bacteria. Here we can see a large amount of peptidoglycan, and some wall-associated proteins are there. And the unique feature we are discussing, that is the teichoic acids are present. When the teichoic acids are combining with the lipid, they are called lipoteichoic acid. And here we can see the cytoplasmic membrane. So this is the cell wall structure of a gram-positive bacteria. On the other hand, this is the schematic representation of the cell wall of gram-negative bacteria, where we can see, in addition to the cytoplasmic membrane, there is another outer cell membrane, just like plasma membrane. There are some proteins and some specialized protein channels. It's called the porine. You can see here a very little amount of peptidoglycan is present, and the special feature is the lipopolysaccharides. Lipopolysaccharide means lipid and polysaccharide, that is carbohydrate. So here the lipid, the yellow color, this is the lipid A, this constant, it is here, and these are the polysaccharide. This is a polysaccharide chain, This is called the lipopolysaccharide or LPS. So this lipopolysaccharide is made up of lipid A. This one is the core polysaccharide, the lower one, and the upper one, that is protruded one, is called the O polysaccharide. So here we can see here, this is the lipid A. It is made up of long chain fatty acid. Then here glucosamine, that is a pyrophosphate polymer. Then the core polysaccharide, and then the repeat units of the polysaccharides, that is the O polysaccharide. So they're acting as an endotoxin or pyrogen. That means they can cause fever. They are toxin in nature. And the structure is a lipid A and polysaccharide. Polysaccharide is responsible for the O antigen of E. coli and Salmonella. Now here we are just comparing the structures of gram-positive cell wall and gram-negative cell wall. So if two bacteria are there, the gram-positive and gram-negative, we can see the cell structure, a large amount of peptidoglycan in gram-positive bacteria along with the teichoic acid, and we can see the lipoteichoic acid here. And here in the gram-negative bacteria, two cell membrane, that is in addition to the normal cytoplasmic membrane, there is one outer cell membrane and a very lesser amount of peptidoglycan remain present. Here we can see the peptidoglycan, and some porine channels are present here. And the lipopolysaccharides, you can see the protruded part of the saccharides and lipid part is embedded. So in addition to that, some periplasmic space is present here. Again, we can see here, this is the gram-positive, so a large amount of peptidoglycan is present, and this is the cytoplasmic membrane. Here in the gram-negative one, we can see a very lesser amount of peptidoglycan, and this is the outer membrane, and here are lipopolysaccharides are present, and one periplasmic space is present here. Now this N-acetyl glucosamine, that is NAG, and N-acetyl uramic acid, that is NAM, they are remaining attached with each other with the help of beta-1,4 linkage, and this bond is lysozyme sensitive. Now this is the structure, the intricate molecular structure of NAM and NAG. So if we can see here, we can see that NAM, each NAM contains 4 amino acid chains. A chain with 4 amino acids. The amino acids are L-alanine, D-glutamine, L-lysine, and D-alanine. So we can see here each and every NAM is containing these 4 amino acids. Okay, now just see this D-alanine, that is the 4th amino acid of this NAM, is making connection with the 3rd amino acid of the previous one. Okay, so 4th amino acid, that is D-alanine, is making connection with the 3rd amino acid, that is L-lysine, of the previous one with the help of a 5-glycine interlinkage. So this blue colored linkage is nothing but made up of 5 glycine molecules. So here in every case we are finding this type of interlinkage, which is responsible for this thickly bound structure of peptidoglycan. So here we can see that it is preparing this 5-4 linkages, this, that is, it is making the 4th amino acid is making linkage with the 3rd amino acid of the previous one with the help of 5-glycine molecule bridge. Now with the help of this we can divide, or we can specify the differences between the gram-positive and gram-negative bacteria. Gram-positive, the main feature is teichoic acid. This presence in gram-positive only. It is made up of glycerol phosphates and ribitol, and it helps in for attachment with the phages. Whereas lipopolysaccharide is found only in gram-negative bacteria. It is toxic in nature. It can kill mice, pigs, even the humans. It can cause fever. It stimulates immunity. It is heat resistant, and O antigen of Salmonella and E. coli is caused by this lipopolysaccharide. Now we are coming to the bacterial cytoplasmic membrane. It is composed of a phospholipid bilayer. At the proteins, we can see here the proteins, and encloses contents of the bacterial cell. It is hydrophobic in nature and acts as a barrier to check the leakage of cytoplasmic constituents. Here water can penetrate, it's flexible, it's not very rigid, and covers the cytoplasm. So here we can see the phospholipid bilayer and the peripheral proteins, the transmembrane protein, and the protein when it is combining with the sugar molecules, it is a glycoprotein, and when it is combining with a lipid, this is the glycolipid is formed when the sugar moiety and the lipid A is found. Lipid is found. Now we are coming to the cytoplasm. It's composed of 80% water and 20% salts and proteins. The DNA inside is circular and haploid. Advantages of haploid DNA over diploid one: it's more efficient, it grows quicker. Mutations allow adaptation to environment rapidly. There are plasmids. Plasmids are the extrachromosomal, self-replicating circular DNA, and it imparts antibiotic resistance. But there are no organelles like mitochondria and Golgi. Nucleoid DNA in the bacterial cell is generally confined to this central region. It is not bounded by a membrane but can be seen by transmission electron microscope only. The genophore is sometimes referred to as a bacterial chromosome. It's a long double strand of DNA, usually in one large circle. It includes most of the genetic material of the organism. Plasmid, as we have discussed, plasmids are small circular DNA fragments found in the cytoplasm that contain codes responsible for antibiotic resistance and other characteristics. Plasmids and the associated traits can be transferred between bacteria, even from one bacterial species to another. Here we can see the circular E. coli genome, and this is a random coil of the DNA, and this is the 1000-fold condensed part of the DNA. The cytoplasm is the internal soup of the bacterial cell, which is bounded on the outside by the cell envelope. The cytoplasm is mostly water, but within it are the bacterial inclusions like nucleoid, plasmids, ribosomes, and storage granules, as well as the components necessary for bacterial metabolism. Ribosomes give the cytoplasm of bacteria a granular appearance in electron micrographs. Though smaller than ribosomes in eukaryotic cells, these inclusions have a similar functions in protein synthesis. And regarding the storage granules, nutrients and reserve may be stored in the cytoplasm in the form of glycogen, lipids, polyphosphate, or in some cases, sulfur or nitrogen. Lastly, we are coming to the endospore. Some bacteria can survive hostile environments, often for long time periods, by bundling their genetic material in a tough internal structure. Endospores can withstand heat, cold, radiation, and lack of nutrition. Actually, it is a resistant structure. It takes time and energy to make spores. Location varies. It may be centrally located, it may be subterminal or terminal. Bacillosynthesis spores can be used in the biological warfare even. So, thank you all for watching this video.